right, so for this last session today, I'm going to go through a few of the more specific design considerations, and hopefully, if the technology cooperates, we'll do some live demos of these designs. So bear with me, this is going to be kind of less formal of a presentation and more going out to the websites and putting data in and seeing how to manipulate some of the things that we talked about earlier for some more specific design considerations that aren't just your basic qPCR assays. So specifically, we're going to do a species-specific assay, one that is meant to distinguish between two different species of interest, one that is strain-specific in terms of specific for a particular viral strain, and then CNV assays. And so at least for the first one of these, I'm going to go live outside of the PowerPoint here and actually put the sequences in and walk you through what we would do to design under these certain considerations. So hopefully at the end of this session you'll understand the different types of design specifications for some of these different applications, including species and splice-form specific qPCR and CNV assays, and be able to identify certain design considerations for the different experimental scenarios that you experience, and be able to adjust the basic qPCR design parameters according to these more specific needs. You'll be able to learn how to do an alignment of sequences, which is often the first step in designing these more specific design scenarios, to identify both unique regions as well as similar regions depending on the context of your assay. And then finally learn how to design copy number variation assays. And so going through this, there are several websites that we'll be using that all have important tools for the design process. NCBI for sequence succession, which we talked about earlier this morning. And then Clustal O Alignment, which is a free alignment program available on the web. There are a lot of alignment programs out there. If you're comfortable with a different one, feel free to use whatever alignment program you have in the lab and you feel comfortable using. I tend to use Clustal, and so that's what I'm going to demonstrate today as an option. And then using this alignment, you'll identify common or unique target regions and use PrimerQuest to put these specific regions into PrimerQuest and get assays that are targeted to your either common or unique sites. And then check for specificity using BLAST and check for dimer and secondary structure using the oligoanalyzer tool. And so this is kind of the flow of how we approach designing a custom qPCR assay from start to finish, from getting your sequence of interest from NCBI all the way through checking your primers in oligoanalyzer. And so you'll see this is kind of the common workflow for each of these designs. So first I'm going to just review some of the general design considerations. And so we've seen this before today, and just some general considerations for designing primers and probes. PrimerQuest will do this for you, but every once in a while you'll come across a design where you need to target it so specifically to a certain region that you find yourself having to design your primers and probes by hand, perhaps. So that's when it's important to keep these considerations in mind when you're trying to design a particular assay around certain specifications. And so in general, we like to keep the TM between 60 and 62 for our primers, and making sure that the deviation between the two primers is no more than a degree or so, trying to keep them as close as possible. In standard assays, about 18 to 30 bases for the primers, although based on some particular sequence contexts, you'll find that sometimes this needs to be a lot longer if your sequence is very AT rich, or somewhat shorter if your sequence is very GC rich. Again, GC content somewhere between 35 and 65%-ish, 50% is ideal. Avoiding those runs of more than 4 Gs. Trying to avoid hairpins and dimers. We want to give the assay the best it can by making sure the sequence context is as ideal as possible if we can design around it. Again, there's going to be those situations where you're kind of stuck with a less than ideal sequence context, and then there are ways to work around that. So avoiding SNPs as much as possible, and making sure that your primers are highly specific for your target. And then the probe, ideally 4 to 10 degrees higher TM than your primers, with a length of less than 30 base pairs, but this can, again, be longer with Zen due to the enhanced quenching abilities provided by Zen. A GC content somewhere in the range of 30 to 80% with minimal runs of G sequence. So if you're especially using dyes in the green range, you want to make sure to avoid putting a G base at the 5' end of your probe. You can get some quenching by G if you place a G base at the 5' end, especially if you're using those FAM hex green dyes. And then the location of the probe can either be on the sense or antisense strand. So if you're dealing with a particular sequence context, know that you can always go to the antisense strand if the sense strand is problematic. And then aiming for amplicon sizes somewhere around 70 to 150 bases or so. So the first important thing is knowing your gene of interest, knowing it in and out. So we talked about how to access that information from NCBI today, but before you begin a design, you want to know what the gene organization is, the exon and introns, and different transcripts. And so again, we go to NCBI to get that information. We see a nice graphical representation of our gene of interest and different transcripts for that gene. And you can use this kind of as an overview if you're trying to design, for example, an isoform-specific assay or an assay that will hit all isoforms. This is a good place to start because you can kind of get a graphical representation of what is similar and what is different about your gene of interest and all of its transcripts. From here, we can obtain our sequences in FASTA format. Again, this is a very useful format for then copying and pasting into PrimerQuest and Clustal or other alignment programs. And so you can take these FASTA sequences for your genes of interest. Let's say we want to compare two similar genes and we want to know which regions are similar and which regions are different. We can take these FASTA sequences and put them into that Clustal alignment program and get a good overview of what regions will be our target regions to design to. So here is what that Clustal program looks like if you go to the Clustal website. From here, you want to select DNA, and then you input your sequences of interest. And you want to make sure to include this caret symbol to separate each of the sequences that you're looking at. You can align 2 sequences or many sequences, as many as if you're looking at several different sequences. And you just need to separate each of those with this caret and a unique identifier. If you copy and paste the Clustal format over directly from NCBI, you can just use it as is. And then you can adjust here if you want to what kind of output it's going to give you. I typically leave it as Clustal, either with or without numbers, depending on what you prefer to look at. And then you push the submit button. And what happens is Clustal nicely aligns our 2 sequences of interest for us, marks gaps here with these hashed lines, and stars indicate similar bases, same base. So wherever we see the lack of this star down here, we know that this region is different between our 2 sequences. And so this can help us if we're trying to design an assay, let's say, for example, that cross-reacts between human and rat, then we can find regions that are highly similar between these 2 and put those into PrimerQuest to design to. Alternatively, if you want a specific assay, you can look for regions where there's differences and design to that. And so that's what we're going to be going through here over the next hour or so. We're going to talk about how to tailor your designs to designing around these specific regions. And so one important design consideration is designing to avoid genomic DNA amplification. And so you want to make sure that if there is, for some reason, a small residual amount of genomic DNA in your sample, that your assay will not amplify that. So you can design to prevent that by either designing your primers across exon-exon junctions, so by placing one primer to span two exons, two different adjacent exons, or you can design your primers within two adjacent exons, but spanning a large intron, and the larger the better. So if you've got a large intron between where your two primers lie down, the likelihood of that amplifying, even if there is genomic DNA present, is slim to none. So you can kind of guard against that in your design. And then, as always, DNase treatment is essential for eliminating potential genomic DNA contamination.
So how do you define large? How large is large?
Well, you've got to ask yourself, depending on your cycling parameters, what is likely to amplify? So a couple thousand base pairs when you're fast cycling, a few thousand base pairs when you're fast cycling is not likely to amplify in the amount of time. If you're giving it an elongation,
a small extension time.
Yeah, it just really depends on the extension time and your
Okay.
cycling parameters. But obviously, the larger intron, the better.
Okay.
All right, so our first example here is a qPCR assay to differentiate between two similar genes. And for this assay, we're going to design something that specifically recognizes our Arabidopsis RCI2A versus RCI2B. So these are very homologous. They're 78% similar in sequence. And so bear with me as I access the sequences and we put those into our clustral program to align them. So I've already downloaded these from NCBI. So I just went to NCBI. You can either find them by this identifier or by typing in your organism and gene of interest. And then we can just copy and paste that information. Actually, you can copy it directly from NCBI as well. All right, so we've got our clustral Omega program open. Switch this to DNA. Copy and paste that second sequence in. And then submit. And it only takes a few seconds before it aligns your sequences. Of course, if you put in more sequences, it takes a little bit longer, but it's still very quick. And then from here, you can kind of scroll through to see what's similar, what's different about these sequences, try to get an idea of regions to design to, and download the alignment file. As a Word file. And that gives you the ability to manipulate it some more. And so there, we can save this. We can highlight regions which look like they're going to be unique or overlapping and try to get an idea. So in this case, we want to design an assay that's specific for RCI2A and RCI2B. So looking through here at A, the region that seems the most different and the best target for assay design would be this 3' region here. So this whole region that's not present in the 2B. So for this first design, that's what I'm going to do. I'm going to highlight this whole region here and input that into primer quest. So for convenience, I have that ready to go there. And now if we go to primer quest, and paste our sequence in. In this case, any assay that falls within that region should be good for what we need. So we can just leave the default parameters as is and select qPCR 2' and Chrome. We're only giving it a very small region, so we probably don't expect a large number of assays to come up, but okay, it gives us one. So now we can now go back and adjust some of those parameters if we want some additional assays to work with. We can start by looking at this one and seeing if this one will suit our needs. And so from here you can click View Assay Details, and it shows us forward primer, probe, reverse primer, and where they're located on that sequence of interest. So going back here, this is our first region we're designing to. The next step would be to blast each of these primers to make sure that they truly are specific for our CI2A. And then while we're doing that, we can check for SNPs, although in this it's not applicable. SNPs are not annotated or available for all species. So for this plant sequence, we won't have to check for SNPs. But if you're working in mouse, human, rat, you want to check for SNPs as well. And then finally, we'll go into Oligo Analyzer to check for dimers and hairpins. So pull up NCBI Blast. Copy and paste in our forward primer. Some ends to separate it. And the reverse primer. So you can do that. If you want to add your probe in, you can put your probe in as well. So for this, we would select others, somewhat similar sequences. And then I like to go down to the algorithm parameters and switch our word size to 7. So hopefully in just a few seconds, all of our hits will appear. And hopefully they'll be specific to our CIA2, 2A. Scroll through these and see an overall summary of the hits. And so now we see this, our Arabidopsis 2B. So that's concerning because we're designing specifically to 2A. So why is that there? What's happening? So we go down here to look further at what that might be. And you can actually click right there and it'll pull you down there. And you see, oh, well, this is a user-submitted sequence, AF, where they've created a fusion between 2A and 2B. So that's not likely to be a concern for our assay design. So look through the rest of these, and you'll see they are all 2A specific. They align on a single chromosome. And so the last check of this is good. So most likely this is going to be a specific assay. So from here, we want to take those primers and probe into Oligo Analyzer. so start with our forward primer. So in looking at these in oligoanalyzer, we want to set the divalent salt and DNTP concentration similar to what is actually going to be in our reaction. So we can change these, and that'll give us a more accurate representation of what the melting temperature for this oligo is going to be. By selecting Analyze, it kind of gives you an overview of the sequence, the melting temperature, percent GC, and such.
Can you just take a moment to show how the TM changes when you encounter?
Oh, sure. Yep. So if you don't change that salt and DNTP, and just put in your oligo as is. So remember the TM was 62 when we did change that. Select Analyze. Melting temperature is 54 degrees. You can see by putting the appropriate salt concentration in there that you get a much better representation of what the melting temperature for your oligo is going to be.
Quick question, so the DNT concentration would come off the label from our DNA polymerase master mix that we buy?
Your DNTP, so usually, is that
Part of the master
is
mix.
it part of the master mix? It's not always available, but we usually, as a default, set to 3 and 0.8 as a good starting. If you know for a fact that your master mix deviates from that, you can set it accordingly.
Yeah, it's a good middle ground to kind of use the default rather than nothing. Rather than have these. But of course, if you know the more appropriate concentration, then that's what you should.
So then we can.
0.8 or 8?
Oh, it's 0.8. It just doesn't. The zero is not showing up very well, or the point is not showing up very well. 0.8. And so then we can click on the hairpin, and so oligoanalyzer will assess the secondary structure for us. You can see down here. These are the predicted secondary structures for this oligo. TMs well below that 40-degree cutoff that we said earlier is kind of where you start to need to look at it closely and be concerned. You can pull this up and just take a look at what secondary structures predicted, but this should not be any problem for this oligo. So then looking at self-dimers. You can see here, this is the most stable self-dimer that this is predicting. And that's, again, well below that -9 cutoff. This is not a very stable dimer and likely won't be an issue for your reactions. So now we want to repeat this. Oh, first we want to do heterodimer check with our other primer and our probe. All right, and so then looking at these predicted dimers, none of them appear to be very stable. So the forward and reverse primer look like they would be a good pair. So now we just want to check and make sure that the probe also doesn't have any potential of dimers, forming a dimer with the primer as well. And that looks good too. So we've gotten our sequence of interest and put it into PrimerQuest. We got the assay from PrimerQuest and we've checked blasted for specificity, and that looked good. We've checked our primers and first combination of primers in oligoanalyzer to look for dimers and hairpins, and everything looks good. Now we'd also repeat that with the other primers as well. And for the sake of time today, I'm not going to repeat that with all of the primers, but you kind of get the idea of what what you'd need to do, you would check then the forward or the reverse primer and probe for secondary structure and all possible heterodimer combinations.
So if we like that, we can click that button up there, what had to order.
Right, so if that assay checks out, then you can, from the prime request, I think if you're logged in,
Well, there's a button on that previous slide.
Mhm.
here we go. So you can select that and add the assay to your cart.
Okay, yes, maybe it's all three.
That will get you both of those primers and that probe. All right. So now that's one assay for this. We wanted something that's specific to RCI2A. We wanted something that's specific to RCI2B. So that all those considerations check out for 2A. Now we go back and look at our sequence again and try to figure out what a good region would be to design an assay for RCI2B. And so in doing so, this region here I kind of highlighted as a potential good place to try. A lot of this is going to be trial and error. You're going to see an area that looks like it would be good to design to, and then start to design to it and need to change things up. And so it's a lot of looking through, finding good areas, trying a design there, and then moving to a different location if you need to. And so for here, we see there are some mismatches between the sequences, especially in this area. And then we get to here where there's a whole region that's present in 2B that's absent in 2A. So it would be nice if we could target our probe to this area right here that's not present. That way, even if we do get some amplification, the probe's not going to be able to lay down over that region that's absent, so we shouldn't get any signal. So that's what I'm going to try first. You can also try lying down one of your primers in that area as well and avoid amplification altogether. And so, again, we're going to put that specific region of sequence that I had highlighted in the blue box. We're going to put that into prime request. And then I've highlighted here in this sequence where that deleted portion was. And that'll be important in a minute when we're telling prime request where we want it to design to. So for this case, you have a couple options. You can take your chances and just increase the number of assays here and go through one by one and hope one of them fits your needs, or you can specifically tailor prime request to kind of nudge it to give it what you want, and that's what we're going to do in this demo. So zoom in. Can everybody see that? Okay. And so we'll go down to this bottom section. All of this can stay the same. What we want to do is target our assay so the probe falls in that particular deleted region. And we can do that here. So by telling prime request, we know, so that's from 116 through 132. So we can tell prime request not to put the probe from 0 through 105, for example. Oh, it's got to start with 1. That's right. And then also 140 through so it shows the entire input sequence here in green. And what we're telling it is we want prime request to put the probe in this little specified area. So by excluding the probe from the beginning and the end, hopefully we're going to target prime request to give us a probe in that specific region that's different. And so it does. It gives us a couple, 3 different assays to choose from. And so now from here, we look at these one by one and line them up on our clustral alignment to make sure that the probe does encompass that region as we intended. And if we would do that, I guess first to point out, you notice since we targeted the probe to such a small region, each of these different assays gives us the same probe. It's varying the primer. You'll see, that it's assay dependent, but we only gave it a few 30 bases to work with or so. And so all of these have the same probe. So we want to check the first of these assays. Here's the first assay. I just wanted to point out here, sometimes customers are leery of using an assay where the probe and a primer butt up right up against each other. We usually recommend leaving at least a couple bases. In this case, since one's on the sense strand and one's on the antisense strand, it would be fine. It would likely work fine. But if you're concerned about that, you can always go to the second assay in the prime request list and go with that one instead. And so in this case, this is the second assay that prime request developed. And so we can see here, again, our clustral alignment. We have our 2A assay that we designed. I've got the primers and probes highlighted here, and then the 2B, that assay 2 from prime request is here. And so you can see the probe lays down nicely over that deleted region. Also, the forward primer has multiple mismatches in that sequence, so likely that forward primer wouldn't lay down very well. We wouldn't get amplification, and the probe definitely won't be able to lay down on the opposite target. So this assay should be highly specific for our CI2B assay. Again, I'm not going to repeat it here, but we can blast each of these for specificity, make sure that does only pick up our CI2B, check for SNPs if applicable, which isn't here, and then go to oligo analyzer, again to check these for dimers and hairpins. There's kind of just the overall strategy for how you would come about designing an assay that's specific for each of these 2 similar genes.
I have a question on that previous slide, on that probe, on the first one, isn't that probe overlapping a little bit on these?
Right here.
Right.
Yep.
But would you want to just not have that part of the probe when you order it?
No, you can order it as is. Likely when that probe tries to lay down, it doesn't have very much similarity on either end. It's not going to be able to lay down. And actually, you can use thermodynamics programs to calculate mass match mismatch of that. What you would see is that the TM for that probe to lay down on that unintended target, it would just
2 less bases on each side?
no, probably wouldn't make a difference. That there's no way thermodynamically that probe is going to be able to lay down.
Well, the bases you have TM of your probe. Because if you end up making a 2 lower TM of your probe just to avoid 2 or 3 bases on the terminal, you may end up compromising that assay. Just remember that even if on this 2 or 3, the majority of that sequence won't sit down on a mismatched target. So it's not going to pick up
Yeah, especially since a single mismatch can affect the TM up to 8 degrees. So if you count in, what is that, like 10 or 12 mismatches there, there's going to be a huge TM difference between those. In fact, I calculated it when I was putting this together. Is it on the next slide? No. I did calculate it, and it was so different that it actually said that the likelihood of that probe sitting on the off target was negative, which is a limitation of the thermodynamics program. But it's pretty convincing that that probe's not going to be able to lay down. All right, so a second design strategy. Now for this one, I won't step out and do everything. We've already kind of showed how to put the sequences and align them in Clustal and use some of the programs. But if there's anything you want me to step out of PowerPoint and demo live, please let me know and I can do that. Otherwise, we're just going to go over a couple other design scenarios and the process that we would go about to design for these different considerations. So in this case, we're going to design a qPCR assay to distinguish between 2 very similar microbial sequences. And so the sequences that I've chosen for the demo are 2 viral sequences. They're from the Helicoverpa, Zika, and Amigara nucleopolyhedrovirus strain, NPV. And so these viruses infect earworms, which then feed on plants and crops. So hopefully broad applicability to a lot of people in the audience. We've got something that hits plants and viruses and insects. But anyway, so these are very similar sequences, and if we want to have an assay that's only going to target either the Zika or the Armagera virus, we can do so. So similarly to before, we go to Clustal, and we input each of our sequences into Clustal and obtain the alignment. And then we can look at that alignment to try to find regions that are similar and different. So in this case, we can see some gaps, and this looks like it would be a good region to design to. This looks like it's a good region to design to. I always start by looking for those gaps, so portions of sequence that are present in one and not the other. There are also regions that just are highly dissimilar. And so for this first example, we're going to select this region here and design to this top sequence so that it's specific for that and doesn't recognize Zika. And so this is that portion of the sequence that I had highlighted in the blue box from Armagera. And this time we're going to, again, show our custom design parameters so that we can target that. We can increase the number of results to return, and we can also target the probe region as we did before to try to force PrimerQuest to put a probe over that region that's different between our 2 sequences. And so in doing so, we get this assay, which we can then use this to overlap that onto our Clustal alignment and make sure that that looks unique, and then take those sequences to BLAST and do a BLAST again to make sure that they really do only recognize our one strain of interest. Again, SNPs are probably not relevant. And then use oligoanalyzer to check the primers and probes, again, for dimers and hairpins. And so this is the assay that PrimerQuest came up with. And you can see it looks pretty good. There's a mismatch in the forward primer, so it's not as many mismatches as in our previous one. But the fact that this probe here is so different in the 2 sequences, this is a very good assay. So we can actually align the sequences as if this deletion wasn't present. So if the probe was to lay down on that unintended sequence, and you can see how many mismatches there would be. And so there's just no way that that probe is going to lay down And so we would not get, even if we would get amplification, because these primers could potentially amplify, we're not going to get any probe signal. Alternatively, if you feel more comfortable, you can try to design your forward primer or reverse primer in this region too. You can force primer quest to do that and avoid amplification. And so then we repeat that with the other viral strain, the Zia. Here I've selected a portion of the sequence up here. We've got a lot of mismatches, and again, a region that's different between them. So this area right here looks like it would be a good place to target our design. And so we put just that region into primer quest. Again, try to...
Can we go back to the previous slide? Previous one. How do you choose how big the region needs to be? You are now choosing like 4 lines. Could you do just 2 lines?
Yeah, absolutely. There's a limit of 120 base pairs that you have to put in.
So at least 120.
Yeah. 80 bases. 80 bases for this. So anything over 80 bases. So primer quest here will accept anything over 80. So if you put in 60 bases and go to submit, you'll get an error saying there's not enough sequence. Please supply up at least 80 bases.
Is there an advantage of choosing larger portion versus smaller portion?
There is in that you have the potential of getting more designs to choose from, and you're giving primer quest more sequence to work with, so potentially getting higher scoring designs. So it's always best to give it as much sequence as you can without compromising specificity. So if you're looking to target a really specific region, then you've got to minimize that sequence. But if there was a lot of difference between the 2 sequences, like in that first example where we kind of gave it that whole big chunk of 3' end that was different, that's the ideal situation where you can give it a lot of sequence context to work with, and then PermaQuest will come up with more designs to choose from and higher scoring designs. And so again, it comes up with an assay for us. When we align that on the sequence, you can see we've got several mismatches in our forward primer, which is great. A probe that most likely will not be able to lay down, even though our reverse primer is identical between the 2. This assay should be highly specific for the Zia strain. And so using Clustal and PrimerQuest, we can use those alignments to tell PrimerQuest where to design assays based on our needs, whether that be species specific or strain specific or common. If you want to design an assay that's going to work for 4 or 5 different species, so you want an assay that's going to work in human, mouse, rat, and monkey or something, you can align all of those sequences in PrimerQuest, select regions that are similar, and target those by PrimerQuest. And so lastly, I'm just going to hit a little bit on designing copy number variation assays. A lot of our customers are doing CNV assays. And so just a few considerations for designing copy number assays. CNV assays are important in assessing copy number variation, and CNV is polymorphisms that can influence the expression of genes around a rearranged region, as shown here. So we have just segments of the chromosome where a section of DNA is duplicated. And this can happen in a multitude of ways, either by duplication of just one little segment once, or duplication multiple times, or duplication of one region followed by a larger region multiple times. So CNVs kind of encompass a lot of different genomic duplications. And so we can assess these differences by using CNV assays. So this copy number variation allows for transcription levels to be higher or lower than can be achieved by controlled transcription by just a single copy gene. And they're being associated more and more with a wide variety of diseases, such as cancer, neurological disorders, and immune diseases. And so a lot of people are looking at CNVs in their samples. And we can use primetime qPCR assays and design them to specifically evaluate the copy number of our genomic DNA targets. And so the principle behind doing this is simply to design an assay that's within a single exon of your gene of interest. So if you go to NCBI and download your sequence information for your gene of interest, you can get single exon information from NCBI and then input that into PrimerQuest. So an example of that start by, so we just go to NCBI, nucleotide, so from here, if we select highlight sequence features and switch this to exon, it allows us to scan through our sequence in the different exon arrangement. So we can select whichever exon we want for PrimerQuest to design to. So in this case, bigger's better, I think, sometimes. So we can highlight this whole region and input that into PrimerQuest and design an assay that would be applicable for CNV. Another way to get this same sequence would be gene, and it's really handy. You can change the display settings here from full report to gene table. And then this will give you all of the exon information individually as a table. So you can see here each of those 5 exons. You can select this and then directly copy and paste it into PrimerQuest. And then again, check those assays for dimers and hairpins, and then you have an assay that would be applicable for CNV analysis in your gene of interest. And so really any of the assays would work for this application. You just want to select the assay that's most highly specific and has the best primer and probe in terms of not having any secondary structure or dimer possibilities. And then one final consideration for designing CNV assays is one of the most important considerations is having a good reference gene, there's a known single copy gene that you can use to compare to. We commonly recommend RNase-P for human, and we have an assay for that, or mouse, either TFRC or TERT. There are different single copy genes for different organisms, but you just want to make sure that you have at least one, if not more, known single copy gene as a reference when you're doing CNV assays. And so that is the end of our design consideration training. Does anybody have any questions about any of these designs or designing in general?
On the CMD, so what would the end results look like for a copy number they had 100 for like 50 copies of? So that would be a lower QT after you do the analysis from the single copy?
Right, you're going to see a shift in your CT.
And one more question on your virus. So you go through there, you did the one primer probe combination, and you found another one. So at that point, I would order the first one in Zen, I could order the second one in Genome.
Yep, so if you want to multiplex
And I
those.
can multiplex, what's the probability of that working right out of the box?
It's very sequence dependent, but
So
as long as you've done all your checks on your assay, you've got a PrimerQuest assay, you've checked for dimers and hairpins, and it looks very specific, it's very likely that it'll work quite well. Sometimes with multiplexing, you need to take and optimize each individual reaction separately. So if you throw everything together and have some issues at first. You can take them separately and optimize by TM gradient or whatnot to optimize each reaction. And there are some considerations for multiplexing, as Arita mentioned earlier, but taking all of that into account, there's an excellent chance that they would both work.
And the last question, does the historical data look like? So if I do a PubMed search and get my primer probes that were 10 years ago, would you start there? Would I just start maybe do this, or put them through this? I mean, you think that 10 years ago would give you different results than if you did the analysis today?
Absolutely, especially with the SNPs. If there's the potential of any SNPs in those primers and probes, it could greatly impact your data. And 10 years ago, that information wasn't available. In fact, NCBI updates the SNP database constantly and very frequently, and I think they're up to 9 million SNPs for human, and that's up from like 6 million just several months ago. So it's constantly being updated, and so there kind of is an expiration date in terms of an assay that was successful 10 years ago may work okay, but why hedge your bets? You might as well start with something that you can control and know that you can optimize